Welcome to all students. Myself, Udara, Assistant Professor of Physiology Department, Faculty of Agriculture and Veterinary Science. Today we discuss about our topic physiological role and agricultural use of plant growth regulators. Mainly fooding and plant propagations, in which use protein compound like butyric acids, naphthalene acetic acids, 2,4 2,4,5-D, used for rooting and plant propagations. IPA produce strong fibrous root systems better than naphthalene acetic acid and 2,4,5-D. Germination and dormancy, mainly gibberelline is a potent germination promoter, and abscisic acid germination inhibitor and gibberelline property. Induced dormancy, it causes by abscisic acids. Breaking of dormancy by oxygen and gibberellins. Fruit and developments. Fruit setting is caused by 2, 4, 5, 3, and fruit size increment in grapes by gibberellic acids. Shelf life increment in fruits and flower caused by cytokinin hormone. Good fruit shape is caused by gibberellic acids plus cytokinins. Parthenocarpic fruits is caused by gibberellins, indole acetic acid, and phenylPAN. Sex expressions, production of male flowers by gibberellins, cucumbers, and production of female flowers by auxin and gibberellins. Auxins cause female flowers in cucumbers and gibberellin cause female flowers in males. Abscission, control of abscissions by naphthalene acetic acid and indole acetic acids. Induce abscissions by withdrawal orthogenesis, it caused by auxin and cytokinins. Weed control use mainly auxins hormones as like after 4D and 2,4,5D. Mainly weed control के लिए like use करा सकता है. Plant organ size, increase plant height mainly causes by gibberellic acids, and shorten the plant height use of triiodobenzoic acids. Increased layering used by cytokinin hormones, example PARP, benzyl immunoglobulins, and triiodobenzoic acids, will increase layering. Antioxidants mainly which cause reduce the translation rate. ABA and phenylmercury acetates, which used as a antioxidant in plant. Papaya later flow by ethaphons and rubber latex flow mainly causes by 2,4-D and 2,4,5-D. Fruit ripening causes by withdrawals and sugar cane ripener, glyphosate, and CCC. Mainly hormones, which major functions were produced. Auxin stimulates cell elongations involved in phototrophy, and David trophies, apical dominance, and vascular differentiations. But it productions meristem of apical buds, embryo of seeds, and young leaves. Cytokinin stimulate cell divisions, reverse apical dominance, involved in shoot growth and delay of leaf senescence. Its major function of cytokinin. Productions of cytokinin synthesized in roots and transported to other organs. Ethylene stimulate fruit ripening, leaf and flower senescence and abscissions. mainly produce of productions of styling, tissue of lining, wrapping fruits, nodes of stems, senescent leaves and flowers. Abscisic acid inhibit growth, stimulate stomatal closures, maintain dormancy. Productions of abscisic acid, leaves, stem, and green fruit. Gibberellin stimulate shoot elongations, stimulate bolting and flowering in pineal, regulate productions of hydrolytic agents in grains. Meristems of apical bud and root, young leaves, embryo. So mainly different plant growth regulators which used for agriculture growth, yield, quality improvements, and different productions. As you know about the different जो plant growth regulators होते हैं उनका जो मुख्य का जो यूज होता है कि वो किस तरीके से mainly अपन यूज कर सकते हैं तो इसके लिए mainly आपको जो प्लांट ग्रोथ है उसका जो कमर्शियल यूज है वो किस तरीके से किया जाता है उसके बारे में mainly आपको पता होना चाहिए कि कौन सा प्लांट है और उसके अंदर फ्रूट की क्वालिटी मेंटेन करनी है आपको या इसकी जो mainly इमेचरिंग शूटिंग है उसको कंट्रोल करना है या इसकी जो mainly जो ग्रोथ है उसको मेंटेन करना है उसके अकॉर्डिंग में भी जो डिफरेंस जो हार्मोन्स है उनका यूज किया जाता है मेनली जो जिब्रेलिन है उसका जो सिंथेसिस है वो प्लांट के अंदर इस तरीके से होता है तो मैकेनिज्म एंड एक्शंस है वो मेनली उसका डिफरेंट होता है इट्स लाइक जैसे आपका ओजिन्स है तो ओजिन्स और जिब्रेलिन का जो बायोसिंथेसिस है डिफरेंट टाइप से होता है mainly synthesis of the enzymes alpha amylase one important function of gibberellin is to cause the synthesis of enzymes alpha amylase in the alluvial layers of the endostomes of cereal grains during germination. These enzymes bring about hydrolysis of starch to form simple sugars which are then translocated to growing embryo to provide energy source. Distributions of gibberellin in plants. Gibberellin are found in all parts of fire plant, including shoot, root, leaves, flower, petal, anther, and seeds. In general, reproductive parts contain much higher concentrations of gibberellin than the negative parts. Immature seeds are especially rich in gibberellins, mainly 10 to 100 milligrams per gram fresh weight. In plants, gibberellin occur in two forms, free gibberellins and bound gibberellins. Bound gibberellin usually occur as gibberellin glycosides, cytokinin and kinetins. Kinetin was discovered by Spook and Miller's 1950 from the tobacco gallus, and the chemical substance was identified as 6-furfuryl aminopurines. Because of its specific effect of cytokinin cell lesions, it was called as cytokinin or kinetins. The term cytokinin was proposed by Bethams 1963. Fairley and Ingors 1966 used the term phytokinins for cytokinins because of their plant origins. Chemically, cytokinins are kinins and they are purine derivatives. Cytokinin, besides their main effect on cell lesions, also regulate growth and hence they are considered as natural plant growth hormones. Some of the very important and commonly known naturally occurring cytokinin are coconut milk factors and zeatins. It was also identified that cytokinins as a constituents of tRNA. Naturally occurring cytokinin can be extracted from coconut milk, liquid endosperms of coconuts, tomato juice, flower, and fruit of virus melons. Protopyrus communis, PR, Humus crassiferi glands, and Lycoplasma esculentus. Cambium tissue of Pinus radiata, eucalyptus regens, and Nicotiana pedicums, immature fruit of Giames. Duclans species and Musa species, female genotypes of Ginkgo biloba, fruitlets, embryo and endostomes of Prunus verisca, seedling of Pisum seedling, root exudates of Helianthus annis, and tumor tissue of Tobacco. According to Spook and Armstrong's 1970, at the latest, least seven well established types of cytokinin have been reported from the plants. Biosynthesis. It is assumes that cytokinin are synthesized in the case of purines in plants. Nucleic acid synthesis. Root tip is an important site of its synthesis. However, the flipping seeds and cambial tissue are also the site of cytokinin biosynthesis. Candy 1965 reported that cytokinins move upward, perhaps in the xylem stems. However, basipital movements in the petioles and isolated stems are also observed. Said at 1966 found that auxins enhance kinetic movements across locations in bean stems. Physiological effect of cytokinins, mainly cell divisions. The most important biological effect of kinetins on plant is to induce cell divisions, especially in tobacco with callus, carrot root tissue, soybean, cotyledons, P. callus, etc. Cell enlargements like auxins and gibberellins. The kinetins may also induce cell enlargement. Significant cell enlargements has been observed in the leaves of Phaseolus vulgaris, pumpkin, cotyledons, tobacco pit cultures, cortical cells of tobacco root, etc. Concentrations of apical dominance, external applications of cotyledons. Root growth. Gibberellin have little or no effect on root growth at high concentration with some causes root growth. Bolting and flowering in many herbaceous plants, the early period of growth, so rosety habit with short stems and small leaves. Under short days, the rosety habit is retained, while under long days, bolting occurs. The stem elongates rapidly and is converted into polar axis bearing flower primordial. This bolting can also be induced in such plants by the applications of gibberellin, even under non-inductive soils. In Hyoscyamus niger, a long-day plants, gibberellin treatments cause bolting and flowering under non-inductive soils. While in long-day plants, the gibberellin treatments usually result in early flowering in soil-day plants. Its effects are quite variable. It may either have no effect or inhibit or may activate flowering. Parthenocarpy, germination of the pollen grains is stimulated by gibberellins. Likewise, the growth of the fruit and the formations of parthenocarpic fruit can be induced by gibberellin treatments. It may causes, example, palm and stone fruits, where auxins have failed to induce parthenocarpy. The gibberellin have proven to be successful. Seedless and fleshy tomato and large size seedless grape are produced by gibberellin treatments as commercial scale. Synthesis, synthesis of alpha knowledge and distributions of gibberellin in plants. Gibberellin are mainly all part of the plant's cell patterns, including shoot, root, leaves, flower, petals, and seeds. In general, reproductive parts contain much higher concentrations of gibberellin than the negative parts. Immature seeds are especially rich in gibberellins, 10 to 100 milligram per gram fresh rate. Naturally occurring cytokinins can be extracted from say, coconut milk, liquid, and from coconut, tomato, juice, etc. Cytokinin, mainly physiological effect of cytokinin cell divisions. The most important biological effect of kinetic on plants is to induce cell divisions, especially in tobacco pit cells, carrot root tissue, soybean, cotyledons, P. callus, etc. Cell enlargements like auxins and gibberellins. The carnitine may also induce cell enlargements. Significant cell enlargements have been observed in the leaves of Phaseolus vulgaris. Pumpkin cotyledons, tobacco with cultures, cortical cells of tobacco roots, etc. Concentration of apical dominance. External applications of cytokinins promote the growth of lateral buds and hence counteract the effect of apical dominance. Dormancy of seeds like gibberellin, the dormancy of certain light-sensitive seeds such as lettuce and tobacco can also be broken by kinetic treatments. Delay of senescence, Richmond laying effect. The senescence of leaves usually accompanies with loss of chlorophyll and rapid breakdowns of proteins. Senescence can be postponed to several days by kinetic treatments by improving RNA synthesis followed by protein synthesis. Richmond and Lake 1957, while working on detached leaves of xanthine, found that kinetin was able to postpone the senescence for a number of days. Flower inductions. Cytokinin can be employed successfully to induce flowering in shoulder plants. Morphogenesis. It has been shown that high auxins and low kinetin produce only roots, whereas high kinetin and low auxins would promote the formation of fruit buds. तो ऑल स्टेंट्स इज लाइक जो प्लांट ग्रोथ रेगुलेटर है उनका जो डिफरेंट यूज है वो मेनली किया जाता है तो मेनली जो प्लांट ग्रोथ रेगुलेटर है उनका जो डिफरेंट यूजेज है वो मेनली अकॉर्डिंग टू डिफरेंट कंडीशंस है कि प्लांट के अंदर कौन सी कंडीशन क्रिएट हो रही है तो उसके सॉल्यूशन के लिए मेनली जो डिफरेंट फिजियोलॉजिकल प्लांट ग्रोथ रेगुलेटर्स है उनका यूज किया जाता है